Okay, wonderful. Last 2 days of the school, how is everyone feeling? Okay, I see some thumbs up. That's nice. Great. Okay, so without further ado, Jacopo with this last lecture on the failures of neutral theory.
Okay, so just to summarize the point where we arrived last time, and while people are coming in. So the last thing we did, like sort of in a bit of a rush, was to look at the other patterns that you can predict with this simple theory or this simple toy model, depending how you want to call it. And I'll summarize it again in some rush, and I say that you can predict property of the dynamics, like the time to local extinction, and you can predict spatial properties, which are related to the correlation, the spatial correlation of the position of individuals in space, and how the diversity increases over time, over different areas. Okay. Now, the point I want to start from is how actually neutral theory fails, or what are the let's say the issues of neutral theory. And there has been, especially in the, from the early 2000 to, for about 15 years, a strong, let's say, controversy of why neutral theory was working, if it was actually a useful model or not, and people were sort of strongly criticizing the assumptions. So most of this debate was about the assumptions, right? What I'll try to do here is to give a sense of why these people got a bit angry about the assumption, but also try to focus more on the predictions, right? I mean, we can have models with assumptions that seems very wrong, but then, we have to judge models based on their prediction, not of their assumption, I believe. Okay, so before I move into that, there are questions on the previous part. Okay, so one reason of why this fails is because we know that species are different, right? This is more about the assumption. But for instance, let's say that you want to describe a tropical forest. What you know is that there are many traits you can measure, right? You can measure how big are the trees, you can measure how big are the leaves, how many seeds are produced, how big are the seeds that are produced, etc., etc. And if you collect these with different species, you see that species are very, very different, right? Then you can ask how much these traits actually are distributed, right? And this was what Matteo was showing here. But for sure, we know that species are different, right? Now, in principle, full neutral theorists would say, okay, species are different, but perhaps these traits don't matter at all, right? So what neutral theory is, if you believe in the assumption, sort of, you take the assumption literally, what neutral theory is saying is, yes, there are all these differences, but this is the same as like coloring species, coloring individuals with saying like, I'll describe the dynamics of blue and red individuals. So all these differences are like completely negligible and don't influence ecological dynamics. Of course, this is hard to believe, right? It's hard to believe that, I don't know, how many seeds you produce and what is the germination rate of these seeds doesn't determine your dynamics, right? And now people try to reconcile these things, like sort of suggesting that there are, yes, perhaps there are differences, but somehow these differences lie on some surface, and then they are sort of neutral. But one point of why people really didn't like neutral theory is because it's sort of saying all these differences, all these things that we measure, that we like, that sort of determine the fact that we see like a tropical forest to be so diverse, don't matter. Okay. And now, I mean, it's not just these differences. There are some differences that actually play a role, right? So, for instance, last week, Gara was showing that somehow species abundance is related to to the effect of seedling recruitment in different neighbors. So there are processes, there are things that you can relate to the abundance that is not just stochasticity, right? That has somewhat a signal that you can measure. Okay. So this is one route that you can take to sort of criticize and sort of make the result of neutral theory problematic, because like you know that there are differences and these differences matters in population dynamics. This is one way. Yeah.
No, I just wanted to ask, how does this pattern relate to the power law scaling that you had shown before from the spatial aspect of neutral theory? Is there some sort of a connection or not really?
I haven't thought about this, but I can think about this while not on stage. No, yes, I think that there might be a relation. I mean, this is something that has to do with also with space, like with local. So probably it's related to Janssen-Cornell effect, which is something that you can imagine to add. It's a good how it's related to the correlation.
Yeah.
I think it's a good question. Yeah, I haven't thought about this. Yes, but there could be like yes.
I mean, when you see things like this, I mean, there's a classic thing about correlation versus causation. So is the statement that you're making here is that in something like neutral theory, you never see some random trait being correlated this way with the abundance?
Yes, that in neutral theory, you will not expect, I mean, let's say you expect that traits don't affect, somehow, in a sense, let's say that you have a given species and you measure all the traits of a given species, right? This is a vector where you say, in this vector, I measure like, I don't know, the size of the plants, the number of seeds, etc. In principle, what an ecologist would say is that the birth rate of an individual of that species depends on what the species is, because these are like some functions of the trait of the species, and actually a function also of all the abundances of other species, right? But for sure, there should be traits, like functions of the traits of the species, right? Now, the idea is when you look at neutral theory, you are somewhat saying that this is independent of I, right? So one way this can happen, I mean, the simplest way in which this can happen is that this does not depend on traits. Another more convoluted way in which this can happen is that this depends on traits, but also depends on all the abundances of other species, and somehow these cancel out, right? And so I would say that the case where this is like does not depend on traits explicitly, so that actually these traits don't matter at all. It's like a strong interpretation of neutral theory. The case where, well, this depends on, let's say, the abundances of all the other species. The case where this becomes independent is what some people would call the emergent neutrality, right? Where somehow the ecological dynamics brings the sort of state of the community to a situations where these are independent. Okay? Questions? But the point is, in general, I mean, the expectation is that there should be some signal, right? Yes?
That there could be traits which can affect the results, like certain correlations or certain assumptions that you, like when you assume that everything is random, for example.
I mean, the point is somewhat one, the main route of criticism to neutral theory came from the fact that if you study like a forest and you study plants, you see that there are differences, right? That plants are not the same, and then somehow you develop a sense in which there are patterns there are, let's say, correlations. There are also, I mean, you can build up causal mechanisms of why this particular species only survive in presence of these other species, right? And you can build up this mechanism, you can understand it, right? And so all these mechanisms that you can study, for instance, looking at pairwise, you can do experiment, et cetera, but that you know that, for instance, a given plant species requires the presence of another one to survive, let's say, and you know that that is the case. In neutral theory, this completely disappears.
No, but you mentioned something about all the variations exist along a surface, which you can ignore. I didn't.
Yes, because I didn't explain. What I said is there are two cases in which you can make these rates independent. One case is when you say, which is like the sort of literal interpretation of neutral theory, is there are no differences. Another way, which I'm sort of doing and waving, is to say, now, in principle, the dynamics of a given species depends on the trait and on the abundance of other species, right? So it's a complex function of all these traits. And you can imagine that somehow, for some reason, the abundances of all the other species by the ecological dynamics arrive to a state such that this does not depend anymore on the species.
It's effectively like random.
And effectively new. So this is like sort of what I would call the emergent neutrality. Okay. Other questions? Okay, but these are, in my view at least, these are mostly, let's say, somewhat critique on, well, not exclusively, but mostly on the assumption, right? They're motivated by the fact that the assumptions are realistic. Then one can ask, okay, is there a prediction of neutral theory that clearly fails? Right? The neutral theory tends to fail when we start to look at large time scales, okay? So last time, for instance, I told you about the meta-community dynamics, okay? And the fact that in meta-community dynamics, you have this rate of speciation, which determines the arrival of a new species, right? And now what basically neutral theory happens is that you can imagine that if you focus on a given species, the species was absent. And then at a certain point, there is the speciation event where the species appears. And then the species is subject to these dynamics, right? Okay. Now, many times when a species appears, it goes to extinction, or there is a time after which it goes to extinction, right? So if I repeat the trajectory, I could see something like this, okay? Now, the point is, one question that you can ask is, suppose that I observe a species with a given abundance, okay? So I observe that a species has an abundance here, okay? What is the time it passes to arrive to that abundance, okay? Is the question clear? So I have many possible trajectories. Well, in principle, I could have, if I observe a given abundance, right? I could have many different trajectories corresponding to different points where the species emerged. And I can ask, what is the average time of this age? This is something that has been studied by the father of neutral theory in population genetics, Kimura. And basically, you have a close expression of what is this time. And there are two important parameters, just two parameters. One, this does not depend on the mutation rate because this is some events that appear in the past. And depends only on what is the total population size, how many individuals there are in total, and X, which is the relative abundance. So what fraction of individuals belong to that species? And then you can say, okay, let's take neutral theory seriously, and let's imagine that we look at a large scale, for instance, the Amazon rainforest, and we ask, what is the expected age of a given species, right? So assuming that the dynamics in the Amazon rainforest is neutral, right? So that what we see different species because we see different abundance because they arose at different time in the past and because they are subject to fluctuations. Okay. So you do this game and you say, okay, I look in the Amazon, I have about n to the 11 trees. Okay, the most abundant tree has a relative abundance of about 1%. Right? This gives a sense of the skewness of this distribution. So you have that you have many, many different species. The most abundant is 1%, which is a big fraction. And then you say the generation time of trees, it's, let's take about 30 years. Then if you substitute over there, so here you get the age in terms of number of generations. So you have to translate it into years. You get that the age of the most abundant species is 10 to the 11 years. And this is almost 10 times the age of the universe. Okay? And you know that sort of you should expect something about 10 to the 8. So this is completely off compared to what you expect. Now, there are some caveats you can build in. The fact that, for instance, this capital N is not constant. So you should not use the population size now. You should use the harmonic mean of population. But you can sort of build up stories, but still you get similar problems. Okay? Yes. So this means, suppose that I take now what I see now, I'm at a given time, which is today, which is okay. Today, what I say is I observe that the most abundant species has this abundance. Okay. Now, what I want to ask is, I assume that the dynamics was neutral. Okay. So somehow I can imagine that I run many, many simulations where this species was sort of born, let's say, a given time in the past, a given time, condition on the fact that today I have this abundance, right? So today I have to arrive here and I have all the trajectories that goes back to some time in the past, right? For instance, I could have a trajectory where this species was born yesterday, and this is a very, 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 very unlikely trajectory. Right? And then I have, as I move, I have more or less likely trajectory, right? Because according to neutral theory, this has to come up with going up and down, which are stochastic. And so I have all these times, right? And I take the average of these times, and what I get is the test. Okay. Is always true? No, there is a distribution, right? And the distribution as that's a good question. No, I think you have a well-defined mode, right? Because it's very unlikely that you have, because the point is that if you look at very, very large time scales, the trajectories will look like something like this, where you can grow and then you collapse. Right? So it's very unlikely to have a species that survive for a very, 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 very long time. Right? So I think the distribution has a well-defined mode. Question? No, you have the microphone. Okay. Good. Okay. So this is one way in which sort of neutral theory, I think, produce like sort of wrong assumptions. And then I'll explain why, sort of how to reconcile this one simple way in which to reconcile these two things. The fact that neutral theory works well for some patterns, but works very poorly for other patterns. Okay. And there are many, people studied many ways in which to do this game and sort of non-neutral extension. So I'll just present one, which is by no means like the most interesting one, is the one I did. So it's easier for me, but so you can imagine that you have like something that a simple non-neutral extension of the model we considered before. And the simple non-neutral extension is a case where you have you have your forest, and then what you assume is that at any given stage you produce seeds, and the number of seeds that you produce, it's species dependent. Okay, and then seeds compete. Okay, so when one individual dies, and then it's replaced by somewhat you create this pool of seeds, and the number of seeds that each species produces, it's species dependent. And then you pick 2 or more seeds at random, and then these seeds compete, right, for the available space. So one tree died. Then there is this space which is free. I select some trees and they compete, right? And I can choose how many trees compete, and they compete according to some rule, right? And the rule is such that where you say, okay, if I have a tree of species A and a tree of species B, the probability, I have a given probability that A wins over B, which is not 1/2. Okay? And then when I reproduce, there is some speciation probability, which is a probability that a new tree arises. Okay? So basically, this is non-neutral because you can have different fertilities, different number of seeds you produce, and you can have different competitive abilities, right? So you can, if you have two seeds of two different species, it's more likely that one seed wins over the other, okay? Now, the case of neutral theory is the case where, I mean, you can see it in different ways, but basically one case is when all the fertilities are the same, and when you pick at random one seed, you pick only one seed at random, right? So it never competes with another one. So, or when the competitive abilities are the same. So the probability that A wins over B, it's 1/2. So to cut the story short, because then I want to move to another topic, what you see is that if you look at the species abundance distribution, okay, if the so what you see is that here it's comparing like models with different number of players, let's say, and you compare neutral theory with models that are non-neutral. And what you see is that species abundance distribution does not depend much on this hypothesis, right? Especially you have regimes, there's a regime where speciation rate is large enough, where basically you don't see a difference, even if the model is non-neutral. Okay? On the other hand, what you can ask is, okay, this is one pattern, then you can study other patterns and you see similar things. Yeah. This is like basically, here we were interested in the number of seeds that were competing. But for the message I want to give now, it's like comparing the red with the other ones. Okay? And so the red is neutral, the no-red are non-neutral versions. Yes, that you have this seed pool, and then you pick like 2 or 3 or 10 seeds, and they compete. Right? Here there is an interesting story about but the point is, if you want, the important things I want you to know about this is that so in neutral theory, if you neglect stochasticity, there is no dynamics, right? The expected value, so if you ask in neutral theory, if you ask how this quantity change over time, in this setting is equal to 0. Okay? So there is no deterministic dynamics. In the case where there are more players, so m larger or equal than 2, this is different from 0, and in particular you have some fixed points which are species dependent. So you arrive really to a community that has some. So there is a deterministic part which is non-trivial. Okay. But the point here is that let's say there are parameter regimes where you don't see a difference in this part. On the other hand, you can ask, okay, what if I look at the age of the species? And then it's where you see a difference, right? So this is the same with the same parameters that I showed you before. What you ask here is, what is the species age as a function of abundance? What you see in neutral theory is that this is like monotonically increasing, and this was the problem I mentioned before, right? That if you have a very, very abundant species, neutral theory predicts that it's going to have a very, very large age. Why? Because in order to arrive to this point, just due to fluctuations, you need a long time. While in all these cases, you don't have this, you have a plateau, because in this case, you have actually a fixed point you are arriving to, around which you fluctuate. And that dynamic is fast, right? You have this driver that pushes you there, and then you fluctuate around this point, right? So the message here, and this is the message I wanted to give with this, is that the success of a theory or a toy model is not something absolute, right? So you can have, it also depends on what patterns you're looking at, right? You can have patterns that you can recapitulate with this very simple model, but then this simple model fails completely and reproduces some pattern. So somehow the success of neutral theory in reproducing species abundance distribution, it's a statement both about neutral theory, but also about the pattern itself, right? That somehow this pattern, it's not informative about processes that can be revealed by looking at other patterns, okay?
How do we see a direct connection between the things that you added to the theory and this particular graph, which addition is resulting in us?
The important addition here is the fact that you have the dynamics drives you to a fixed point, right? So somehow the trajectory don't look like this stochastic going up and down, but there is, when a species appears, let's say, when a species first appears, there is a push to arrive to some state, right? And then it's going to oscillate about these states, and the oscillations are large enough that they look like the oscillations you have in neutral theory. But if you look at the time for large abundances is dominated by this. Okay, so somehow a slightly better way to say this is, in this mode, in neutral theory, if you see a species with large abundance, it has a large abundance just because by chance it arrived to that large abundance, and so that requires a long time. If in this model you see a species with large abundance, is because this species is fluctuate around a fixed point which corresponds to large abundance. And if you fluctuate around a fixed point which corresponds to a large abundance, it means that there is a force that pushes you there. And therefore the time, it's not particularly, you don't need to have a especially large age, okay? Yes.
Can we think of neutral theory as like a null model that sort of explains some patterns like abundance distributions, which can happen even in the absence of the differences between species? And then when you see deviations from that null model predictions, then you sort of incorporate other ingredients?
I think you can see neutral theory as a null model. It's not necessarily the null model, right? In the sense that it's something that I think has the merit of producing predictions and has the merit of being wrong, right? Which, as the title suggests, it's a good thing, right? So these things, I think it's something that makes neutral theory worth to look at, in the sense that you can say, okay, this fails because of this reason. So it's a null model. It doesn't mean that in any case is the reference point you should compare your system to, okay? Because somehow when you consider a null model, you are saying, I'm separating of what is null to what is interesting somehow, right? And so you're saying I have a null mechanisms, which is in this case is demographic stochasticity. And then I want to see what is deviating from the demographic stochasticity, right? It's not obvious that this is like the good reference point. But perhaps what I'm going to say next clarifies this.
The probability distribution for trajectories is, how do we relate this to the straight line graph? I earlier asked about—
This one?
Yeah. The red line is just straight. So.
The red line, it's exactly this thing. So I don't need to go back, but remember that I wrote something like A of x equal to -2 n x 1 minus x log x. Okay.
Yeah, so I was asking, how do we relate it to this particular question of older trajectory? It need not be necessarily true that older trajectories are abundant species have more probabilistically older trajectories.
No, this is true for neutral theory. So more abundant species. So this is this is exactly what that curve is, right? And you see that for large x, this becomes something that is 2n x log x. I mean, here you don't see the log x correction, but there is a log x correction. So this is exactly what that plot shows. Okay. Other questions? Yes.
It's like as the abundance increases, it's probably like dipping downwards. Is that going to keep going downwards, or what's happening there?
I mean, we were thinking about why it goes down. I don't remember anymore the explanation. I mean, one point is that you're looking at relative abundances. So if you have a large relative abundance, it means that you have low diversity, which for some reason I think it's related to why it goes down. I think it's probably because if you have a very large abundance, it means that you are a sort of, I mean, in this process, you have also the rise of these mutants, etc. So it might be that these cases where you have a very large abundance, when you take along, are things that are very superior competitors to others. So it takes for them a very long, so basically they don't coexist with others, okay? So they are sweeps that like basically drive to extinction many other species, and then you have some diversity due to stochasticity, but basically they're superior to others, and so it takes a little bit less of time to go. While in this case, in the middle, it's like they coexist with others, so perhaps the dynamics is that. But this is like a sort of story I'm partially making up now. Okay. Yes.
So in neutral and non-neutral theory, the abundance distribution doesn't like matter. We always get this.
Yes.
So I was trying to connect it, but in neutral theory, the abundance is high, but for small spaces, because it's very unlikely that while fluctuating, you will go to a large abundance. But in non-neutral theory, you fluctuate around your fixed point. Then also the getting high abundance is rare. Is it because like getting a fixed point at very large abundance is rare?
I mean, it's, okay. Yes, I would say so. Yes. I mean, the point is that also when you look at the species abundance distribution, sort of the bulk of the distribution are rare species. I mean, a species with low absolute abundance. So the shape of this distribution is dominated by species that are rare. And now species that are rare are species that are at low absolute abundance. So where stochasticity matters a lot.
Yeah, I think I was thinking of something similar. So I don't know too much about this model, of course, but in the standard sort of non-neutral things in population genetics, I think the reason why you have this deviation is because, of course, for every species, you have a period where it has sort of stochastic dynamics and a period where you have nearly deterministic dynamics, right? And so that like how big that period is in your abundance depends on how competitive or fit you are, right? So I'm guessing in this case, what happens is that you have some non-neutral thing that makes you better. So you have some initial period in which you're nearly stochastic, but then if you cross it, then very nearly deterministically, you both go to large abundance, and then you're not, you can have a low age because you're doing it nearly deterministically. Fluctuations matter less and less. So I'm feeling like that might be the general intuition.
That's correct, and you can look at it in the reverse instead of looking at the trajectory for individual species. If you take a snapshot and you look at different species, most of the species are still in these regimes where they don't fit the deterministic dynamics. And so when you look at species abundant distribution, you're mostly describing this state. Well, when you look at these, you are picking up explicitly the species.
Exactly. So for the lucky few that escape this, then their age can be anything because they kind of deterministically nearly deterministically went to large abundance.
Good. So I will move on to the next part, if it's okay. But now this requires me to understand when. Okay, here. Okay, So what I want to do is to do like a sort of now a story in parallel, somewhat in the same spirit of neutral theory, but explaining something that it's in some sense different way of modeling fluctuations in communities, still with the idea of trying to reproducing patterns. Okay. So the point is, again, going back to this slide here, and what we did before was focusing basically on one column of this matrix, right, and focusing on species abundance distribution, and that was the way I was motivating neutral theory. What I want to do now is to focus on this matrix and the fact that it's noisy and sparse. And somewhat the goal of what I want to describe now is the following. So suppose that you go around and you measure this matrix, okay? So you have one of these metrics. The question is, what is the ensemble of this matrix, right? You will have a matrix where you have entries, and in general, you can imagine that this comes from some counting, right? So you will have, I don't know, species 1, 2, 3, 4, and then sample A, B, C, D, E. And here you have 3, 45, 0, 50, 3, and here you have 4, 7, 0, etc. Right? So you have this matrix where you have counts, okay? And perhaps, I mean, the counts can also be, total counts can be different. And I want to ask is, suppose that I want to write a simple program that generates matrices that are statistically similar to the ones I have I observed. Okay? The underlying idea is somehow, since these are I mean, in principle we have other data, which here I removed, but let's say that these are the data that we have. Okay, if I want to have a model or a theory that reproduce this kind of data, it should at least reproduce like this sort of salient statistical features of this matrix, right? So I don't want to have models that predicts that this is a tree. So I don't want my ecological models or my ecological theory to say, oh, this is really tree. What I want to do is that what I get here is something that looks statistically similar, right? You can play different games with this, right? So, for instance, neutral theory tells you a matrix, right? And the matrix that neutral theory gives you is a distribution of these elements where species are exchangeable, right? So in neutral theory, and this is like the sort of key statistical feature of neutral theory, in neutral theory, the probability that species 1 has abundance N1, and let's say species 2 has abundance N2, and species 3 has abundance N3, this probability is equal to having N2, N3, N1. Okay? In neutral theory, the probability is exchangeable. Okay? The process is exchangeable. You don't have like, and this is, by the way, is exactly the reason why it's analytically tractable, and people did a lot of things about this. Okay. Okay. So the setting where I'll explain this is the setting where I'm mostly working on, which is the setting of microbial communities. So where these species are somewhat defined microbial populations, and the samples are different samples. Okay. So just to have you so that you have an idea of how this works. So basically, you have some environment, and so how these matrices are produced in the context of microbial ecology. So you have an environment. What you do is that you take a sample of this environment. Okay. And then what you do with this sample is that you extract DNA, okay, and then you amplify particular region of this DNA, which corresponds to the RNA associated to ribosomes. And then you basically look how many different sequences you have, and you use these sequences to construct populations. Okay, so these populations are sometimes, I mean, the way in which you label these different sequences. There are different ways. Can be like sometimes you can encounter this name ASVs or OTUs, which are basically way of clustering or not clustering these sequences, right? So the idea is I have many different cells that are different to each other in an environment. And then I take a very small part of their DNA, okay? And then I look at this soup of these little parts of DNA. And then I say, if this little part is more similar than a given amount, this is the same population, okay? Is this clear? So the point is, there is, it's not like going around and, I mean, I'm also simplify that, but it's not like going around to the forest and say, oh, this is 3A, this is 3B. Here there is like an experimental pipeline with many assumptions and many caveats. And there is a bioinformatics pipeline of assigning these sequences of cleaning these sequences and assign them to populations, okay? But for my purpose here, what we're gonna have is that at the end, we are gonna have a matrix like the one that Matteo was describing, okay? Where we'll have different species, which we know comes from these, and different samples, which can be the same, I mean, some of the same community between codes at different times, or could be different communities. And then we'll have abundances, and some species could be present, other could be absent, okay? So from this moment on, I will forget where the data come from somehow, and I'll focus on the metrics that we have at the end. Okay. Now, this is something that some slides that I add and that resonates with what Matteo was saying, is just to say that we expect the dynamics of this community to be complex, and the reason why it's complex is that what determines the composition of a community are many environmental factors. So you remember in the 3 tables that Matteo was showing, there is the environment, right? And this environment is fluctuating. And there are many environmental factors. So these other metrics is high dimensional. And then it's because like the physiology and the genetics of these organisms is complex, right? Because there are many traits. This is the other metrics, right? So somehow in focusing on these metrics here, we forget that different samples are different. There are reasons of why these different samples are different. There are drivers, and we are not considering them now. And different species are different because they have different traits, okay? And so the approach here is, okay, so we have all these things. How do we deal with these? The idea, as I said, is to focus, sort of forget about these things, because it's, I mean, for many reasons, are not easy to measure, or it's very hard to deal with this high-dimensional structure, and we don't have a way to measure all these traits and all these environmental factors, and ask, okay, let's focus on what is the statistical structure of these abundances. Okay, so this is like a recap of what we did last time, which is a very simple way to look at this data, right? It's basically asking what is the distribution of columns here. Okay, so what is the species abundance distribution? So just to recast what we saw previously in this kind of setting, the species abundance distribution, it's basically asking, suppose that I take a column here. So I take a given community. What I'm going to ask is, what is the probability distribution of observing a given set of abundances, right? So in this case, it's the cumulative distribution. So it's what is the probability of observing a given species that is observed with a given number of counts, a larger number of counts larger than k. Okay. Now, one thing that it's, I think, very simple to explain, but it's not that let's say that's well stated often, is that species abundant distribution puts together two very different sources of variation. And the two sources of variation that it's sort of conflate together are a variation across communities and a variation across species. Okay, so you can imagine that you can obtain a curve like this, so a distribution of the elements of some type in two very extreme cases. Right? One extreme case is a case where let me start from an extreme case is where species don't fluctuate across environments, across communities, but they have different abundances. Okay, so this will be, for instance, the result of a deterministic dynamics corresponding to different fixed points, where you run the dynamics all the time and you get always the same result. Okay, or they corresponds to a case which is neutral theory-like, where actually species fluctuate a lot, but they are completely exchangeable and identically distributed, right? So the idea is that when you have something like this, the sort of a species has nothing, there is nothing about the species that determines the fact that it has an abundance. It's just one random drawn from a distribution. And what you have in a sample is that you have this variation, but if you take another sample, it's completely shuffled. Okay, so somehow, and this perhaps connects with what you showed before, somehow the species abundant distribution sort of puts together these different sources of variation. Okay, so one question that you can ask is, can I try to disentangle the two? Right? And an obvious way to disentangle the two is to ask, instead of looking at the distribution over columns, to look at the distribution over rows, right? Yes. Sorry, the distribution over rows instead of.
The other example that was the one on the right, that also does include, like, going back to your earlier version of saying that the weak sort of neutral theory says that there are traits, but there are a bunch of interactions that wash out those traits. So it does, the one on the right also includes the one on the left to an extent, right?
Well, I mean, you can imagine, I'm not sure of the questions, but these are two extreme cases, right?
Yeah.
So you get that this in general, and we're going to see that, the variation that you have here, right, it's a combination of these two sources of variation, right? So you will be always somewhat in the middle. What I'm saying is you can imagine two extreme cases, right? So this case here is that you have only variation across species and you don't have variation across communities. And this case is the case where you have only variation across communities and no variation across species. Okay. The point is, when you do this game of going from a sample, extracting DNA, sequencing, etc., what you end up having is a given number of sequences of reads that corresponds to a given species. Okay, so to a very sort of base extent is I go to a forest and I don't count every tree, but I start counting tree at random. At the end, I will have a given number of individuals I assign to a species. Right? This is a discrete number. I could say 0 is 1, is 10, is 45. Okay, so it's this entry here, which is a number. This is just the cumulative probability distribution of a column, right? You say, how many, what is the fraction of these, of the species that have abundance larger than 0? Larger or equal than 0, the fraction is 1. How many have a fraction of, how many entries have a number of reads, an abundance which is larger or equal than 1? It's going to be something smaller. How many? So it's the cumulative distribution. Okay. If you want, it's very much related to the rank plot. Other questions? Okay. So basically, if you're thinking of a time series, so where these samples are like sort of collected in time, what I'm asking, if I look at the distribution over columns instead of over rows, is if you have these fluctuations, what is the distribution of these abundances? Okay.
Time here, time, is that samples?
You can do both. I'll show you data on both cases. So you can have data where the columns are, let's say, you look at the gut microbiome of an individual, and the different columns are different times at which you took the data
From the same
individual.
Same individual.
Yes. So I
Gut
look at yes, I sequence your gut microbiome, and the sequence is today, tomorrow, 2 days from now, etc. Right. And so in that case, you have a time series.
Okay, that's time.
Another situation is I take like these people, and then I'll sequence the microbiome of us, of everyone here, right? In both cases, I will have at the end a data structure, if you want, that looks like that. But clearly, in one case, I have like a time, and in the other case, I have. Okay. Okay. So blah 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 blah. Okay. So what you obtain if you do this game is that basically, I mean, I'll tell you the result and then tell you that there are some machinery to get these pictures. So basically, the result is if you look at the distribution across rows, this distribution is very well described by a gamma distribution. And this is true both if you take the gut microbiome of different people over time, okay? And if you look at different samples from different ecosystems. So you take like, I don't know, 100 different individuals and you look at the gut microbiome. You take 100 rivers and you look at the microbes living there, okay? So the first point is, the sort of result I want to tell you is this distribution, the distribution that describes the fluctuations on these rows, the fluctuations over time, is gamma distribution. Okay. Now, in order to obtain these, there are like some technical caveats. So one point that you have is that when you do this game of sampling, right, you have a source of noise which is due to sampling, at least, right? So if I go around and take random individuals and assign them to species, there is a probability that I never see a given species, not because it's not there, but because I didn't sample it, okay? So there are sampling errors, and the sampling errors, of course, change the shape of this, change the distribution. Another thing is that if you look at individual species, you have like limited amount of samples, so you have like a lot of fluctuations. So what this shows is an aggregation over species. So it's what is the typical fluctuations. And in order to have aggregation over species, you have somehow to rescale them. Okay. So this is each of the colors corresponds to an aggregation over species. Okay. Now, this is a way to visualize the distribution. You can do things more carefully. So you can do like, you can say, is this distribution more gamma or more log normal, right? How much can I really say it's gamma? You can say, okay, I'll compare it with another distribution, right? In statistics, you can never say what is how good a statistical model is. You can only say how bad it is, right? So you can only compare it with something and say it's less bad than something else. Right? And for instance, if you compare with a log normal, which would have like some sort of symmetric shape, you see that this model does significantly better for most of the species that you have. Okay? You can do other way to test this. The point is, it seems to be the case is that these fluctuations are well described by gamma distribution. Okay? So this tells us that somehow, if you want to characterize a row here, these entries here, the sort of the true abundances that generate these fluctuations are gamma distributed. Okay. The risk, I don't know if I want to take it under from, because like in some sense this is important to get this figure, but it's more I mean, in this figure what I said is that you have to rescale these abundances because you want to compare different species, right? But sort of why you need to be this, you have to the problem of stories is that they're always linear. But the reason why you do something in these linear stories, it's because you knew something that I haven't explained yet. So basically, the idea is that these are gamma distributed. So, okay, why it's going back? Good. Now, what you know is that, okay, you have different species and their fluctuations are well described by this distribution. But that's what you can ask is, okay, these fluctuations, if I have a gamma distribution, which is, by the way, I haven't said that, it's something like P of X, it's proportional to some X beta minus 1, e to the minus X over some parameter, if you want. Okay? So this is how this distribution is. What you can ask is, okay, this distribution has two parameters, which I can recast in the average and the variability. And what I can ask is, okay, in principle, different species could have different averages, right? And so I can ask, how are the average distributed across species? So do species have the same average or not? Okay. And if you do that, you find another pattern, again, with some rescaling. But the pattern you find is that the distribution of this average is well described by a log normal distribution. Now, the point is that the things is under the carpet, and then I'll take it out a bit, is that you don't see the full distribution because of these sampling problems, right? You are sampling out of the community a finite number of individuals, but you are sampling a finite number of sequences. So if you have a species which is very, very rare, you might never observe it, okay? So what you see is that you see a distribution that looks like a log normal with a cutoff. And this cutoff is determined by how much you sample this community. Okay, yes. Average over communities, right? So you have, imagine that you have this row. And then you say, I look at what is the average of this row. And then I have another row, which corresponds to another species, and I look at the average of this. Yes, yes, exactly. Good question. I mean, in some sense, this is like doing it blindly, right? I'm saying, let's look at what I see, and then let's try to think about why I see it. Okay. Sorry. Yes, the rescaling is just because. So in this case, the rescaling is just because you want this distribution to collapse to a standard normal.
So that has nothing to do with the,
no.
life history of the generation.
No.
Thanks.
Okay. This is the second thing you see. And then you can ask, okay, now I have some means and I have some variability. Right? How is the variability related to the mean? Okay. And what you see is that they are not independent. So if you look at the variation of relative abundance, so the variance of this scales has the square of the mean, meaning that the coefficient of variation of this distribution, which in this case is related to this parameter here, does not change with the average abundance. Okay, so most abundant species and species with low abundance have the same coefficient of variation. Okay. Is there a question there? No, okay. Now, this distribution is not there is some variability of this coefficient of variation, and this is like a measure of this coefficient of variation. And actually, you see another pattern also there, that somehow there is a distribution of this coefficient of variation which is conserved across communities. So basically, what you have at this point is that you have these three main patterns. Okay, so the fact that you have fluctuations that are gamma, well, the fact that you have species that have an average abundance, which is log normal, and then you have the fact that the variance is proportional to the mean squared, and then the fact that the fluctuations are gamma, that allows me, these three ingredients allow me to generate a matrix like this. Okay? So how do you generate a matrix like this? Yes. Yes, exactly. Exactly. I mean, there is like a caveat there, which again, it's under the carpet, about the fact that you have always this effect of sampling. And so you have a finite number of sequences you want in each sample. And this finite number of sequences depends on the sample for technical reason. So it's not just the distribution of the row counts, but it's what is sort of underlying the distribution of the row counts if then you add this effect of sampling. Okay? So the idea is taking the sampling on a side, if you interpret this as something related to the abundances of the species. So across samples, a given species has a gamma distributed fluctuations. And then the mean and the variance of this distribution are related. And then if you look at different species, so basically, you know that this is generated by gamma distribution. Then the mean and the variance are related. So you need to know the mean to, as a first approximation, know the variance. And then you can ask, okay, what is the mean of a given distribution, a mean of a given species, that it comes from a log-normal distribution. Okay. So basically, if you take these three things, you have a way to generate these matrices. Right? So in particular, what you have is that you can say, if I want to, I can have an ensemble for this matrix, right? I can write what is the probability to observe, if you want, n number of reads, n number of individuals, condition on having capital N number of individuals I sampled, and let's say a given average abundance of a given species. Okay. And then I also know that this average abundance is log normal. So if I integrate out, I know what is this P of n given n. Okay. So some sense I can tell you what is important, the important contribution of a species in this matrix is the average abundance. So condition of an average abundance, I can tell you what is the probability that this number is 3. Or I can tell you if I take an entry at random, what is the probability distribution of this. Okay. And then you can say, good, so then I can use this generated metric to compare patterns, right? And basically, this is like parameter free at this point, right? I mean, it's parameter free in the sense that I use the data to determine the parameters. So I use the data to determine what is the distribution of this mean and variance, and what is the coefficient of variation. And then with this, I can generate data and compare them with the empirical. So this is the number of reads, so it's the entry. So is this n. And then you have the sum over all these entries, which represents the total number of individuals. This is n. Okay. And so for instance, this is how many species you observe as a function of the total number of reads. Right? And so the points are the empirical data, the line is the prediction you get. I mean, it's like that you express as an integral, right? It's a closed form expression based on the parameters you have determined. And this works very well, both in determining the trends and determining the typical number of species you observe in a sample. You can look at species abundance distribution. So the first pattern I showed, which have this shape, and you see sort of the effect of having communities with more or less individual samples. You can look at what is the probability of observing a given species in a given number of samples. So the fact that species can mean 0 or non-zero in a given number of samples. And you can ask, if I have a species condition on having a given average abundance, I don't know, of 10 to the minus 3, in how many samples do I expect to observe that species, right? And this is okay. Questions? Yes.
This is very cool. Just trying to understand better. So if I were to construct the matrix, a non-negative matrix of this kind without any insight, what would that distribution be? Do we have a sense of?
Yeah, I mean, this is exactly the question I asked myself, right? So these are the data you have typically, right? So what is the ensemble of this data, right? I mean, without, at this point, without like trying to find any mechanisms, right? But what is the statistical structure of this data that we want, right? In a very, if you want, without invoking any process before that, right? Just what is the statistical ensemble? I mean, neutral theory gives you one, right? For instance, neutral theory tells you these are exchangeable, right? So you should not expect a difference, a big difference here, right? Other questions? Yeah.
Yeah. So maybe I misunderstood. Is the beta, the value of beta that you get from the data, right? There's a common value of beta that you get, right? Is that correct?
So I'll show you here. So you get a similar value, but the point is that beta, I mean, let's say, so for some of you have a variability of this beta, okay? And the variability of this beta is not that important to reproduce many patterns. But this variability of this beta is present and you can quantify it. And it's like basically reported here. So this sigma square, it's something related to beta, okay? And what you see is that it's sort of exponentially distributed. This is something, I mean, you have something that it's related to beta, which is exponentially distributed. So this something else, it's related to a parameter of something else actually related.
So beta is not fixed, but it's exponentially distributed. Is that what you're saying? Sort of.
The point here is not fixed across species, but the variability of beta is independent of the variability of the average abundance. Okay. So somehow you have two parameters. So if you want, empirically, you have the average abundance X, okay? And then you have the variance. Then you can take the coefficient of variation, okay? This quantity and these quantities depends on the species, okay? But if you look at the scatter plot of these two across species, they are independent. Okay? And for many patterns, the variability of this quantity here does not matter.
And you are using that to generate the actual data, right? I mean, that
for the actual data, I actually assumed that this was a constant. So I was neglecting this variability. So it's not really needed.
So do you have a closed form expression for that Pn, Pnn?
Yeah, it's very simple. So this one, it's I mean, condition on the capital N, it's a negative binomial, because basically it's the gamma distributed abundances multiplied by, I mean, convoluted with a Poisson because you sample. And this is like something more complicated because you have, it's basically the convolution of this with a log normal. Yeah. Is in the notes that I'll share. Now there is a broken link. It's done on purpose, but it's going to be repaired after the lecture. So there is all the sort of calculation on how to derive these patterns in the notes. Okay. So for instance, I mean, just to give you an example, how do you calculate, I don't know, this one, okay? How many species do you expect to observe? The number of species you expect to observe is 1 minus the probability that a species is absent, right? So the number of species is proportional to some total number of species you could possibly have, which is this one. Well, it's related to that one, times the probability that a species is not absent. So that pattern, you see, that is like this quantity here as a function of capital N. This is an example. Okay. Okay. So this is about statistical properties, and now this is sort of gives a good starting point to capture the statistical properties. There are also statistical properties that are not reproduced this way. So for instance, if you start looking at correlations across species, right? You observe distribution of correlations. You see that species are, the distribution of correlations you see is different from what you get in this model. Because in this model, species are sample independently. Okay? But let's say for many studied macroecological patterns, you observe a similar trend. Okay? Now the next step, and I'm gonna do this in the next 11 minutes or 18. I don't know if I should 15 an average. Okay. So it's like to think about dynamics. Okay. Now, how do you think about dynamics? Well, there are, I mean, first of all, what kind of model do you want to build to describe this? Right? There are many choices. And we had a full tree of shown yesterday. What I would argue is that in the data I showed you, there is some sort of similarity in patterns between variation across communities and variation over time. Okay. So the idea is, when you have this kind of similarity, what you're tempted to do is to put variability in somewhat time and think that different communities are different realization of this process. Okay. And so the set of models I'm going, and also in this simple way that I described here, I was describing something where I sort of neglected or forget about species correlations at this stage. So perhaps what I want to start from is something where I describe a single species. And you can interpret the description of a single species as not describing a particular species, but describing the dynamics of a typical species. Right? In some sense, in the same way Mathieu was describing yesterday the idea of dynamical mean field theory, but here it's like without having a microscopic processes at the basis. Like just, let's say I want to describe what is typically happens for a species. And so the type of models you can write is something like this, right? You can say the dynamics of the abundance of a species is some deterministic force, okay? And some stochastic force, okay? This is a very general setting. And it's also very specific, right? I neglected many things. For instance, I said I can meaningfully write this as an effective model where I have a single species. Another assumptions that I took that, for instance, was not clear at the time where I took this, but it's clear now that Mathieu spoke is I decided this is like does not have a delay in time, right? I could imagine that actually the dynamics here could depend on the abundance with some delay in time. This will be a more complicated model, right? But this is a sort of simple setting in which I want to stay. And now the question is, okay, what I want to do is to, I have to specify what is this f of x and what is this d of x, okay? Is this clear, this setting? There are some symbols that I've not described, and I'm sort of assuming that you know what they mean. So I'm assuming that you know that X is an abundance, and I'm assuming that you know that C is a noise. Was I assuming correctly, or it's confusing for someone? Okay. Now, how do I choose this? So one thing that I want to impose, defining this model, is that at stationarity, this model produces a gamma distribution. Okay? Because it seems like it is an observation that I have from the data. Now you can, this model is one-dimensional, is fully solvable, so you can find, so what does it mean at stationarity? It means that P star of X of this process is a gamma, okay? Now these one-dimensional equations can be, the stationary distribution can be exactly solved, and so you can have an expression of the stationary distribution as a function of the deterministic force and the stochastic forces. Right? And so if you want it to be a gamma distribution, you have basically some constraint on how to choose d of x and that of z and f of x. Right? Now, not surprisingly, you cannot fully specify these two terms to get a given stationary distribution, right? You have many choices of them in combination that gives you the same stationary distribution, okay? Now, if you look at simple choices of these d of x and f of x, you end up with these two simple models that you can actually have ecological interpretation of these simple models. You have many more, right? But there are somewhat two that are interesting to look at. So one model is a model we saw there, and it's a model where you have birth. It's a model where the stochasticity comes from demographic noise. And so you have birth, death, and migration. And if you write the sort of effective equation of this model, you get something like this, okay? So you get something where you have a noise which depends on the square root of x, right? Because the noise is demographic. And then the deterministic depends on some linear term which corresponds to a balance between birth and death, and some terms that corresponds to the effect of migration. Yes?
Should there be like
No, in this case, there is. I'm assuming that all the things are independent. No, it's not. These rates are constant. The point of this is that all these rates are constant. Okay, and so you can call this B, D, and M, right? And these parameters that appear here, so tau, x star, and sigma, are function of these parameters. Okay. Then you have another models where I wrote so where you have self-limitation and fluctuating environment, okay? And in this case, what is self-limited growth? Well, basically, you have logistic growth. And then what is fluctuating environment? You have environmental stochasticity, okay? Where this is an environmental stochasticity because this is proportional to x, right? Yes. Here, x star, no, so in this model it looks like, so for demographic stochasticity, x star over tau is equal to the migration rate, and 1 over tau is equal to death minus birth rate. Okay, so you can, yeah, so you can, yeah, it's summary scaling, right? Yes. Yeah, so, so the, here it's like highlighted, but I don't know if you can see the difference. The difference here, it's why these two models are different. So in this case, you have an x over the square root, and here you have an x that is not under the square root. And here you have a term which is, well, you have this linear term, but it's like, here you have a constant, while here it's multiplied by x.
But what is the biological intuition behind having the x under the square root?
Yeah, is exactly, I mean, it's exactly these two processes, right? So in this case, the idea is you have birth, death, migration process, right? And the fact that you have this term is because the sort of deterministic solution, the deterministic part, is determined by the balance between migration and the fact that you have a death rate larger than the birth rate that push you towards zero. And the stochasticity comes from demography, the fact that these are finite individuals, where in this case, the sort of deterministic terms comes from the fact that you have logistic growth, and the fluctuations to the fact that the growth rate is fluctuating. Okay?
I'm not getting the intuition behind how x is being inside the root or outside the root.
Okay. The sort of a precise answer to this would require well more time than 8 minutes and 41 seconds. But yes. Yeah. But yeah. No.
Okay, okay, it works. I should not shout now. Okay, so I think if you remember the derivation yesterday, the G square term, the stochastic term, was actually proportional to the density, right? So that basically means that's an example of demographic stochasticity. So where so the effects of finite size of the population plays a role in stochastic terms. So the equation I derived, which we haven't yet analyzed the stochastic part, right? The stochastic part, has this, functional form of root x, a root rho in the notation I was using. We haven't derived the right side part. So that was just a quick comment on the origin of demographic stochasticity.
Yes. So a very simple intuition I can give, which I don't know if serves as an intuition and simple, is that when you have demographic stochasticity, the noise is at the individual level. Right? So each individual has a finite chance of reproducing or not. So when you look at the population level, which is what we care, this is like the sum of independent noise terms, independent across individuals, right? And so if you sum independent variables, then the amplitude of this sum scales as the square root of x. This is demographic. Environmental is to say all the individuals are subject to the same fluctuations of the environment, right? And so the noise at the individual levels are perfectly correlated in a population. And therefore the sum it's proportional to the size. Yes.
Maybe if I can say something complementary to this, I think the environmental stochasticity is actually easy to see why X dependence gives you that, because generally you'll have exponential growth as X dot equals some R times X. And if you add some noisy thing, which also has an X dependence, now effectively you have X dot equals R plus some noise times X. Right? So it's as if you have an effective R, an effective growth rate that itself is fluctuating. The easiest interpretation of that is that the environment is fluctuating, which is why your growth rate is fluctuating, right? Do you agree, Jacopo?
Yes, it's exactly to say like this. Yes. Okay. Now, okay, I have 6 minutes. How do we distinguish between these two models? Well, there is a first thing we could do, which is saying, okay, we know something. I mean, we can have expectations of what matters for microbial communities, right? So for instance, for microbial communities, we have the expectations that their populations are very large, right? 10 to the 9 individuals per milliliter, let's say, as a proxy. And if you have very large populations, in order to have demographic stochasticity to matters, you need to have sort of very large values of sigma, which corresponds to a sort of fine-tuning of these parameters. So you need to have really that the birth and the death rate are both very large, but both very, very close, okay? So it's sort of, it puts you, in order to have, when you have very large number of individuals, in order to have large fluctuations due to demographic noise, purely demographic noise, you need to go to a very sort of deep corner in parameter space. Okay? This is one thing. Another thing that you can ask is, well, now, for now, I talked about species being independent, but you can say, well, I know that species are not independent, but if you have demographic stochasticity, I mean, by definition, by construction, demographic stochasticity is something where the noise at the individual level is independent. So what you would expect is that somehow, on a end-waving way, if demographic stochasticity is the source of fluctuations, similar species should not be more correlated than distant species. Okay. While if environmental fluctuations are the main source of noise, you can expect that similar species are influenced by the environment in similar ways. And this is what you observe in the data, right? That you can look at the fluctuation, the correlation of fluctuations of similar species, and you see that species that are close in phylogeny, they are typically positively correlated, okay? But this is a very indirect evidence, and is not an evidence at all, because like I'm starting with very simple models, and then I'm using these very simple models to say, yeah, demographic stochasticity or environmental fluctuations, but you might have ways where due to interaction, demographic stochasticity actually produce some sort of correlations, okay? So the path I want to follow is to say, okay, let's look at dynamics in 3 minutes. And basically, if you look at dynamics, what you have is that you have 2 different models, okay, that have different transition probabilities and the same stationary distribution, okay? So the question is, how do you distinguish them? Okay. One way to distinguish them is to ask questions that are explicitly dynamics, right? So one question is to ask, you remember the RI, so if you look at a given time leg, for instance, 1 day, you ask, what is the distribution of the jumps of abundance? Okay. And in the model of for people interested in doing calculation and reading books of solution of Fokker-Planck equations with all sort of methods, in this case, the solution is actually analytically obtainable. And you get that the sort of fluctuations you find is something like this, where you have this tent shape distribution. While in this case, for environmental fluctuations, there is no closed solutions, and you get something that is actually very simple, which is Gaussian, okay? So that these jumps are Gaussian-like, which works remarkably well, I mean, more than what you would expect. Now, then what you can do is that you can look at data. Now, there is a caveat in looking at data, which becomes very important when you look at this data, which is due to the fluctuations of this term here. But I'll put it under the carpet because I don't have time. And what you obtain is, if you normalize the data well, is that you obtain that this distribution is Gaussian. Okay. So these are 3 different distributions where what you do is that you average over species. So you look at the distribution of these jumps over time, averaged over species. Okay. And the caveat is in this averaging over species. So this is like 3 examples from 3 different individuals from 3 different data sets. And another way to look at how much something is Gaussian is to look at kurtosis. And what you see is like a set of different trajectories. And you see that it looks like around 3, so Gaussian-like. And actually, if you do things like more carefully, doing model selection, blah, 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 you find that there is 2 data sets where you have actually more non-Gaussian species than Gaussian ones, okay? Which are these 2 individuals. And so you can ask, okay, why? So, well, if you look at the distribution, then when you aggregate, you see that there is like trends that are non-Gaussian-like, that you have this more wide distribution. Oh, no, I'm done. Now, one of these 2 data sets, it's like something very specific. It's like the time series following Salmonella infection. So somehow it's interesting that we find this difference following a big perturbation. And the other case is a little bit less trivial, but to cut the story short, basically you can say that there are, in this trajectory over long times, you see unstable species, right? You see, and we have a way to detect that I will not describe here, but basically you see that the trajectories of species look like something like this, and then you can have jumps, right? You have a few of them. It's not obvious where they come from. They come from in groups, and the number of such these jumps that you observe across different individuals is different. And these 2 cases are 2 cases where you have many of these jumps, right? So where this is the percentage of species that don't show these jumps. So here you have a big percentage of, a larger percentage of jumps. And then if you say, okay, let's remove these jumps, etc., you find something that is more Gaussian than before. Okay, I'll conclude the story here. So I went very fast in the last 5 minutes, but the idea is what? Is that you observe this pattern, and then you can sort of use these patterns to inform very simple ecological models with captures, which includes some very simple processes, which can be migration, birth and death, self-regulation, environmental fluctuations. And you can use properties of the data to distinguish between the models. And the way you go about distinguishing the models is use this as a baseline and not try to prove the model right, but try to prove the model wrong, right? And somehow these very simple models, you can imagine them as a lens, right, into reality. So you look at the reality under the assumption that this model is right, but trying to magnify the deviation, right? And in trying to magnify the deviations, for instance, you discover things like this that then are not predicted by the model, but they underlie some sort of non-trivial dynamics, okay?